present some of the application of in-cell NMR that I think now start to be relatively popular, and a lot of people are enthusiastic about in-cell NMR. It's still a niche technique, and there are not so wide application, but I think it's very, very vital, important, and I hope I'll show you the relevance of this approach for characterizing biological processes, biological molecules, biological processes, drug screening, and so on. And the aspect that what we developed in particular, because in-cell NMR, I think is known, means high resolution NMR of macromolecules inside the living cells. So you put living cells in the NMR tube and in the NMR instrument, and you collect the NMR spectra. There are different ways to have labeled proteins or labeled nucleic acid inside the cell, and one very effective that I developed in Florence is just to have the direct expression of the proteins of interest inside the cell that we put in the NMR instrument, because this is allowed to characterize the process as from the beginning, and we don't require heterologous expression. The proteins are directly expressed by human cell. And I'm characterizing human proteins, human processes in a human cell. And just to show this is a relatively old example, but the application, for example, on the production and characterization of the maturation of superoxide dismutase, which is an enzyme which goes through a series of post-translational modifications, require the binding of metal ions, dimerization, the disulfide bond formation to stabilize the structure. And so then this can be done only if you have the protein directly produced in the cell. When you produce in coli and then you import, obviously it's already folded, you cannot follow this process. And so this is an example just to show you the quality of the spectra that we can get. And so this is human SOD that is in living human cells. And then you can, if you use labeled cysteines, we can even easily monitor the oxidation state of the protein. And so then for this specific protein, we characterize a number of conditions and factors affecting the folding and the maturation and the different behavior of mutants that are related to ALS pathology. And there is a striking different behavior in the cell with respect to in vitro. And what I would like to present, this is a relatively old story. What I would like to present is more how it can be exploited, this approach to characterize binding of small molecules. And in the specific case of the mutants, the pathological mutants of SOD, they tend to monomerize and so then to aggregates. And there have been proposed type of drugs, molecules, which stabilize the dimer through a bond, a bridge between two cysteines, the two cysteines of the two subunits. And so these are a number of molecules we have characterized. And then, for example, interesting example is this epsilon, which is already a used drug. And this is binds indeed these cysteines and bridge this. But if you put this in presence of glutathione, which is present in the cell, SOD is present in the cytoplasm, this epsilon is released because the bond is broken. Okay, this is in vitro. What is happening in the cell? Totally different behavior, because if we add the epsilon to the cell, epsilon is able to diffuse to the plasma membrane and to bind to the protein, to the cysteine, and stabilize the the structure, but also, as I said, these are all in cell, on living cell spectra, so that I hope you appreciate the quality of the spectra. And for example, if you take this mutant is a pathological mutant of SOD, and in absence of epsilon, this mutant doesn't fold, and so it's floppy and tend to aggregate. Is not structurally stable. If we add epsilon, this favor the formation of the native disulfide bond and stabilize the conformation of the protein. So we have a totally different behavior in vitro and in vivo. So one take-home message is that if you want to characterize some biological process, you need really to verify which is the behavior of that protein, that those biomolecules in the cell. Then you can deeply analyze it in vitro, but as long as you are able to obtain the same status that you have in the cell, otherwise is meaningless. And another example, and the main story I would like to present is the usage, the exploitation of in-cell NMR for drug screening. That is common to use NMR for screening molecules, fragment, and so on, and can be ligand-based or protein-based, target-based. And this approach specifically is target-based. We can do ligand-based as well. And we choose one substrate that is carbonic anhydrase, is a 32 kilodalton, and is a target of drugs already on the market for a number of diseases. Some are in development for cancer, and there are drugs against carbonic anhydrase 2 against glaucoma. And this is a zinc enzyme, and the drugs, what they do, they bind to zinc and inhibit the activity of carbonic anhydrase, that is the one of hydrating CO2 produced using the respiration. So this protein, despite the size, it provides a reasonably well-resolved spectra in the cell. And then, I mean, this is unfortunately small because of the slide, but if we enlarge it, then we can resolve and verify all the assignment, and this is the structure is maintained, and then we can follow what is happening if we expose the cell to drugs. And also what is relevant in this specific case has, as I said, that the drug binds to zinc, and zinc is coordinated by the histidines, and the protons of the side chains, the NHs of the side chain bound to histidine, are shifted outside the large envelope of the protein. And so then you can see that we can have very high, well-resolved proton, 1D proton NMR, very old style, very simple, but this can be used for drug screening. So we can use just the classical HMQC, so fast HMQC, that is, we can acquire in a relatively short period of time, but also even faster, in 1-2 minutes, we can acquire this very nice 1D spectra. And then you can see that these are the two drugs which are already approved, already since years on the market, that they produce a marked difference in the NMR spectra as they are added to the cell. And so then these are sulfonamide as type of molecules. And so then we analyze a series of sulfonamides. And I would like to call your attention on these two specific molecules, 1 and 2. And if you read the inhibition constant or the affinity constant is exactly the same. But look at that number 1 is, molecule number 1 is binding to the active site, so it is in the cell, while number 2, it doesn't bind. doesn't enter the cell. There are molecules that are very similar, that have exactly the same thermodynamics, the same affinity in vitro, and they have totally different behavior in the cell. So this is just the first point, that is how effective is this approach for the early stage of drug development, because immediately you can check whether some molecule can pass the plasma membrane, enter the cell, and decide that you can implement and so on. We can go further, and we can analyze the dose dependence effect, how much is bound as a function of the amount of drug that is added to the cell, and how much time cells are exposed before we acquire the spectra. And we found that the rate limiting step is the diffusion through the plasma membrane. And from these measurements, we can determine the permeability coefficient, and we can correlate this with the potency of the drug. And we were very glad to find that for the 2 approved drugs, which have essentially exactly the same affinity constant, in the prescription, you need 10 times more of one main component of the compound with respect to the other. And the permeability and the efficacy that we monitor is exactly the same, one drug with respect to the other. So this is really an approach which can have a lot of applications in the field of drug development at the early stage, and would be very effective before going in more detailed study, really clinical studies, because this gives information at atomic level and on the kinetic. And I have other aspects, because we can do also real time. And one of the issue of the challenges in cell NMR is to follow real-time processes. And the problem is that the cell inside the NMR tube, they last for 1-2 hours, because there is no oxygen, there is no nutrient, and so they finish them. And so then to address this issue, we implemented a kind of a microbioreactor, starting from the Bruker Insight equipment, where there is a flow of oxygen and nutrient. And then we added a coaxial membrane, which allows, is better seen here, which allows exchange. I mean, the cells are outside, and through here, the inlet, there is oxygen is flowing and nutrient is flowing, and then they pass through the membrane to the NMR tube where cells are there. And also there is exchange that the metabolites can get rid of. And also there is the issue of maintaining cells in suspension and not to sediment. So there are a number of technical points. We found that the Percol is the best, and we agarose to make just the plug, the bottom plug. And here we monitor the lifetime of cells, and they can stay, we check about for 40 hours, so then we stop, and then we easy way to check the viability of the cell through NMR using phosphor 31, because ATP formation is the indication of the viability of the cell. And so this is a very good equipment to do real-time binding of the drug. And here you see the difference, as I show you, as I mentioned you, of these 2 different drugs. This is, as I said, are already approved, are already in the market. That one, you see that there is a slow change in the NMR spectra, while the other is really, really fast. And so then here, these are the difference in the change in the spectra. And then you can do through some statistical analysis, you can measure also the kinetics of the uptake, the diffusion in the cell and the uptake by the target. And we use this approach to characterize a series of approved drugs, not only against carbonic anhydrase, but also against other targets, because this is also another important point to know, to check whether approved drugs, even if they are targeting a different substrate, if they can be sequestered by a different protein. And carbonic anhydrase is relatively abundant in the cell, and usually we try to keep the level as low as possible until the detectability of the NMR spectra, and in some cases almost with each the physiological levels of other proteins. So here is a series of molecules, also sulfonamides, that are inhibitors of other targets, and all of these are approved drugs. And then we can follow the different behavior when cells are exposed at different dose and with different times. And when we see that there are, for some of these drugs, the inhibition, the binding to carbonic anhydrase is immediate. Some other is really slow. And so on time average, we see that there is some are fully bound, some are only fraction. And then we can follow this. But what is, we can classify this are that there are some classes that are fast diffusion that they're diffusing, and so they bind as soon as the cell is exposed, and other are slow diffusing. But then there is this third class. There are these four molecules that have a peculiar behavior, because they initially bind to the target, to carbonic anhydrase, and then slowly are released. And so then they are released. And also there is a different behavior whether the cells are exposed to the drug, so then external to the cell there is the drug, or the drug is removed from the sample. And so then with this approach, we can really characterize all these aspects, including the kinetic aspect and the amount, the level of exposure, and the presence of the drug or not. So then also for pharmacokinetics is a very relevant approach. Another development, but that is unfortunately only really early in progress, fluorinated amino acid. They are very, very relevant, as everybody knows here in this audience, for drug screening, and in particular for in-cell. If we have fluorinated amino acid and we are able to incorporate in a selective way, then obviously, essentially, there is no background and it's easy to follow the process. So with the approach I developed on this direct expression of the protein, we have been able also to incorporate fluorinated amino acid, as we did for selective labeled cysteine, then is selective fluorinated amino acid. And then we can see that the spectra, this is starting work, this is really in preparation, so this is new data, but you see that there is a very good efficacy in the binding. And with this, I finish what I want to say, what I have time to say, because I would have loved to say many other things, just to show the CHERM Center, that is a core center of the Instrat ERIC European infrastructure, and access is available to users and also from overseas users, not only European users. and partially is cover the access. So within DesignNest discovery project, up to 20% of service NMR time provision is covered by the European Commission also for non-European. So if you are interested also to use already the 1.2 gigahertz instrument, as I said yesterday, is running up and running since 2 years, you can apply for this. And then just a quick announcement of the Chianti workshop. For those who knows about this, Rieco has all the t-shirt of this with this nice logo. We will resume, and so then we'll have in June this year. And with this, I thank you for your attention.
Thank you very much, Lucia. That's it gets at the whole issue. We're faced with in bio-NMR. Ultimately, someone says yes, but is it like in the cell? Questions?
Very beautiful, inspirational work. I hope I didn't miss this, but could you tell us a little bit about the concentrations of proteins that you can get through overexpression in the cells, and how much do you need so that you can see your protein above the cellular background?
Yeah, what we can see up to MQC spectra up to 30 micromolar, we can get a good spectra with a 3 millimeter probe, 3 millimeter tube, NMR tube. So this is with the Shigemi tube, so just to reduce the amount of cells, and with that concentration, some of these spectra are slightly higher concentration of the protein, but we have seen that going down in the concentration up to 30 micromolar, we can see the signal. And how to say it, the way how we, if you want a kind of selective label, is because we grow the cell in the rich medium, and as we have transfection, we change the growth medium. Here is with fluorinated amino acid, but with a uniform label. And then if the production of the protein is relatively fast, then we have enough protein in a short period of time, and the rest of the other components of the cell have not incorporated the label isotopes. And the second point, to have a very good background, we subtract the spectra of the background, because what is good is the X cell are absolutely reproducible. Sometimes even we don't collect the background again because we can use one of the year before. Coli is really random, is really difficult, impossible to subtract because it's different every time. Even if you use exactly the same growth condition, EC are exactly always the same.
Thank you.
Really very impressive work. I have a quick question on the general question people ask: why do you need to do in-cell NMR vis-a-vis a lysate NMR? I'm just wondering if there's a way for you to tell whether the protein is in the cytoplasm or the nucleus, because this makes a huge difference for things like transcription factor that move around. Is there a way to tell that?
I mean, first of all, is lysate, the behavior sometimes totally different. So in some cases, proteins we know that are expressed in the cell, we don't detect. As we break cell, they come detectable, even without any purification at all. And also the status of the proteins is different. The binding of the metal is different. Simply, even washing out the cell, but the metal ions are present in the cytoplasm. If the cell is intact, this is control. When it's broken, it's all random. That is the point. The nucleus, I should say that we try to extract a nuclei, and because I did in organello NMR on extracted intact mitochondria, and that is doable. nuclei I try to extract, but the nuclear pore allows too much exchange between the two compartments. So sometimes we separate this and do some gel analysis to check. In some other case, we know that the protein is mostly in the cytoplasm and the volume is much larger.
Yeah, it'll be nice if we could get some sort of a PRE agent either in the cytoplasm or specifically in the nucleus, so that we can see the protein go between the two.
Yeah, that is another point. We are also working in tagging the protein, and then we need another way to incorporate the proteins in the cell. Yeah, yes, that is a good point.
Well, quick question.
I was just wondering about the unstable binding compounds, and I was wondering whether they would just be degraded by the cell. Is that a possibility? And the second thing is, do you have a way of looking at the compounds in your cell that you can monitor their integrity?
We have, in some cases, we did the mass spec after. So then because the compounds are not so high concentration that is all of them easy to be detected with all the background. But we did the mass spec of the lysate in that case, and that were not degraded, at least these molecules.
Thank you.
No, you are welcome.
Well, thank you. That's very inspiring about getting closer to real biology. Let's thank Lucia again.
Thank you.
Okay, our next presenter is Yusuke Okuno from the NIH. And I believe we're going to hear something about co-solute protein interactions. So.
Hi, my name is Yusuke Okuno. I'm from the Grow Group from the National Institute of Health. Today, I'll be talking about some of the new application of the paramagnetic relaxation enhancement to study how small molecules interact with the proteins, with particular focus on the protein folding aspect. So I'd like to first start out with the introduction of the topic I'll be discussing today, which is called Kosovan effect on the protein folding. And Kosovan effect is essentially the phenomenon where addition of small molecule can perturb the protein folding equilibrium. For example, there's a class of molecule called chemical denaturants, such as urea, that actually denatures proteins. So the equilibrium is shifted from the native state to the unfolded state. On the other hand, there's another class of molecule called protecting osmolyte, and that can actually help proteins to fold and stabilize to the native state. And this includes small molecules like glycine butanes and the TMAO. So you can actually control the amount of native state or unfolded state you want by adjusting the concentration of these small molecules, denaturants, and the protecting osmolyte. And this is nothing new, and this kind of observation has been known for many years. But what we don't know, even now, is how does it do that? Like, why just simply adding these small molecules change the protein folding properties of the proteins? And there are many different hypotheses to explain this. And there seems to be like a little consensus on this. And it is when the small molecules happens to denature proteins, it involves some kind of attractive interaction between the small molecules and the proteins. On the other hand, the small molecule tends to stabilize the proteins, that involves some kind of repulsive interactions. But really the exact mechanism of how these small molecules interact on the surface of the proteins in the molecular detail is still largely missing from the literature. And the major reason why that's the case is this interaction between the small molecules and the proteins are extremely weak. So take urea, for example. In order to denature the protein by urea, you need to add molar concentration of urea. So you need to add a lot of molecules. In other words, each individual urea and protein interactions are very, very weak. And it's too weak so that it's invisible to most of the biophysical techniques that include NMR spectroscopy. So most of the experimental data we have about this topic is coming from the thermodynamic experiments, where the observables are like volume, pressure, like concentrations. These are all like helpful, but these are all macroscopic observable. And again, so how does these small molecules interact at the molecular level is largely missing. And also, interestingly, urea not just denatures one specific proteins, it denatures almost any kinds of proteins. And the question is like, how does urea know that it's folded state or unfolded state? There must be some key properties in the native state or unfolded state that allows this small molecule to decide, oh, okay, I'm gonna denature this protein, or I'm gonna stabilize this proteins. So then the question is, what is these properties? So the goal of this talk is to answer some of these questions. But again, the challenge is, how do we study such a weak interactions? And actually we found one exception to this using NMR. And this exception is when the small molecules happen to contain unpaired electrons. And we call this special type of molecule called paramagnetic solutes, and examples are shown here. And what's so special about this paramagnetic solutes is that it contains an electron spin. And the electron spin is just like nuclear spin. It's source of the magnetic field. And if you consider the nuclear spin as like a little magnet, then the electron spin can be considered as like a very strong magnet. So if you could imagine this solutes moving around the proteins like this, it's like saying the strong magnets are moving around the proteins, generating some fluctuating magnetic field. So this field is sensed by these nuclear spins on the proteins, and it's gonna change some of the property of the nuclear spins that include relaxation rates. And in fact, if you add this paramagnetic solutes to the protein system, the relaxation rate significantly increases. Therefore, the name solvent paramagnetic relaxation enhancement, or solvent PRE for short. And a key point is that no matter how weak the chemical interaction is between these paramagnetic solutes and the proteins, we can still detect substantial solvent PRE. So we can measure the solvent PRE regardless of how weak the interaction is. And a couple of years ago, we developed the methodologies to use this solvent PRE data and then get the quantity called R⁻⁶, which is related to the potential of the mean force and the interspin distance between the electron spins and the proton spins like this. It's a little bit complicated, but what it is basically it's the average of the inverse distance between the electron spins on the co-solutes and the nuclear spins on the proteins. So what that means is, let's say co-solutes have attractive interactions on the proteins. Then this co-solutes wants to be close to the protein because it's attracted to the protein, right? So the average distance between the co-solutes and the proteins are short. And since R⁻⁶ is inverse of the interspin distance, therefore R⁻⁶ becomes large. On the other hand, if there's a repulsive interaction between co-solutes and the proteins, then the co-solutes wants to be away from the proteins. And the average distance between the co-solutes and the protein becomes longer, therefore R⁻⁶ becomes small. Therefore, we can quantify how attractive or how repulsive the interaction is between the co-solutes and the protein through the values of the R⁻⁶. And we can get this experimentally by measuring the solvent PRE. So using this new technique, we studied this model protein called DARKNSS3. And DARKNSS3 is a very interesting protein because even in the absence of any chemical denaturants, this protein remains in the native state about 40%, and 60% of the proteins is in the unfolded state. So it's kind of unstable proteins, and the exchange between the native state and the unfolded state are in the slow exchange on the chemical shift time scale. So when you take HSQC, for example, you see two distinct peaks, one coming from the native state and one coming from the unfolded state. So we can measure the solvent PRE on both native state and unfolded state at the same time. And we also studied a particular paramagnetic solute called 3-carbomoyl peroxyl, or 3CY. The reason why we like this molecule is because it turns out it actually denatures this SS3, as shown here. So basically, in the absence of any 3CY, the folded fraction is 40%. But as you add about 200 millimolar concentration of 3CY, the folded fraction becomes 20%. So basically, it denatures these proteins. So the idea is by studying how this 3CY interacts on the proteins on the native state or unfolded state, we want to get the insight about how this chemical denaturation mechanism work. And this is exactly what we did. And again, so shown here is the R⁻⁶ for native state and unfolded state as a function of the residue number. And this is all coming from the amide proton interactions. And just to remind you, the larger the R⁻⁶ is, the more attractive the interaction is. The first immediate observation was that if unfolded state R⁻⁶ is much larger than the native state. So this already tells us that this 3CY preferentially interact on the unfolded state rather than the native state. And the second observation is that some regions on the native state have high R⁻⁶, which is comparable to the unfolded state. So, and these regions are all located at the disorder regions on the native state, more disorder and more solvent exposed regions on the native state. So with this observation, we hypothesized that this 3CY probably wants to interact on the disorder and the more solvent exposed regions on the proteins, which sort of intuitively makes sense. And we wanted to get further more insight about this, how electrostatic interaction play a role in this chemical denaturations. And then we measure so-called effective near surface potential, which is pioneered by Dr. Iwahara, who's giving a talk later in this session. So I'm not going to talk about it in detail, but what it is basically ENS allows you to get a measure of how electrostatic potentials are distributed over the surface of proteins. And we can get this experimentally by using basically measuring the solvent PRE for two different co-solutes with different charge. In this case, I used the negatively charged co-solutes and the neutral co-solutes to get this ENS. And shown here in this plot, the blue circle and the red circle is the ENS that's coming from the experiments. And the black line over here is the theoretical prediction from the Poisson-Boltzmann theory. In both experiment and the theoretical prediction, we saw immediately that the native state has some wide variations ranging from 0 to -40. You see like a zigzag patterns. On the other hand, for the unfolded state, we see this kind of variations are not less pronounced, and it's sort of like homogeneously distributed around -20 millivolts. And this sort of makes sense because unfolded state are less structured. It can take many different conformations. So the charge center that is probably located at the side chain is flexible enough that it can take many different locations on the protein surface. And so basically, when you take the average of many different conformations unfolded state can take, then this charge distribution sort of averages out to some constant number, which happens to be -20 millivolts. So the take-home message is that for the native state, since it has a well-defined structure, some part are charged, some part are not charged. So you see a heterogeneity in the electrostatic distribution, while unfolded state is sort of like homogeneously distributed. So we see two different patterns. So I'd like to conclude this part of this research by answering some of the questions I asked earlier. So how does this small molecules interact at the molecular level, and how does this small molecule distinguish between the native state and unfolded state? Well, what we saw is that this small molecules, the 3CY, preferentially wants to interact on the disorder and more solvent exposed regions on the proteins. And obviously the unfolded state are more solvent exposed and disorder than the native state. Maybe this is the key property that makes this small molecules to decide to know which one's native state and unfolded state. And furthermore, the electrostatic profile seems to be quite different too. Again, the native state has a very heterogeneous patterns, while the unfolded state is homogeneously distributed. So maybe this profile is important for the properties to decide which one's forward state and unfolded state. And for this particular case, for the SH3 and the 3CY interactions, we saw a lot more attractive interactions on the unfolded state than the native state. So we proposed that the mechanism of this chemical denaturation caused by this paramagnetic solute is by stabilizing the unfolded state rather than destabilizing the folded state. So this is basically the summary of my my finding. But lastly, I just want to briefly talk about the research I'm doing right now. So all the data I showed you just now is interaction between paramagnetic solutes and the protein. But what we are really interested in is how the small molecule in general like diamagnetic co-solutes, interact with the proteins. And we came up with this idea to use paramagnetic co-solute as a probe to study how small molecules interact in general. And this, this is coming from the simple competition experiments where we take the solvent PRE in the presence and absence of the small molecules. Take urea, for example. If urea actually binds on the surface of the proteins and covers the surface of the proteins, then this paramagnetic co-solutes have a hard time accessing this surface of the proteins. So it's going to change the way they interact on the proteins. And this change should be manifested in the R2-6 value. So if you take the difference between in the presence and absence of this urea, for example, this delta R2-6 should contain some information about the degree of urea to protein interactions. And to illustrate this point, we did some preliminary experiments on this model peptide called AAMA, and we measured the solvent PRE on the methyl groups in the presence and absence of the glycine butanes and the urea. Just to remind you, the glycine butane folds proteins while the urea denatures proteins. So these two small molecules act in opposite directions, and then see what happens to the delta R2-6. And surprisingly, even at high concentration, like 7 molar of urea, I didn't see that much of a change in R2-6, which sort of indicate that there's a negligible interaction between urea and the methyl group on this peptide. On the other hand, there seems to be increase in R2-6 as I increase the concentration of this glycine butanes, which probably is an indication of the repulsive interaction between the glycine butane and this methyl groups. But this is all like very qualitative interpretations, and we're working on how to interpret this delta R2-6 more rigorously right now. But I'm very excited about this technique because if it really works, then we can probe any kind of small molecule to protein interactions, and we are not really restricted to study co-solvent effect. We can, it might have potential to study other problems such as how drug interacts all across the protein surface. So I'm very excited about that. So with this, first I'd like to thank the organizer for giving me this opportunity to present my work at this wonderful conference. And then I'd like to thank my PI, Maria Sklar, and my group members. And then I'd like to thank all the collaborators and then the audience for listening to me. So thank you very much. So I'll take any questions.
Thanks very much, Yusuke. It really gets at some cool protein biophysics. Questions?
I have a little question here. I'm a little confused by why you actually think there's some kind of specific interaction with your protein. It just seems like you've opened up the protein and made it solvent accessible, as opposed to having some kind of specific interaction with your paramagnetic label. I mean, I can explain all your data just by saying I'm more solvent accessible.
Yes, exactly. So and actually,
So all you've shown is it unfolds the protein.
no, so I mean, that's a good point. Thank you for asking. And actually, so the mechanism I proposed, it's been proposed over probably like 50 years ago. Okay, but all this evidence for this this hypothesis are mostly coming from, again, this thermodynamic data. And basically this work shows, I mean, it's the direct evidence of how this small molecule interact at the residue level, basically molecular level information. So you might say that this is just a, sorry, not this one. This is just a validation of their hypothesis, and you're right. But this is basically the direct evidence, while others are coming from either computational work or the thermodynamic data. So these are sort of like indirect experimental evidence, but this is more direct in that sense. So yeah, that's that.
A quick comment here. In principle, could you get something like a radial distribution function like they do for liquids? Like when you define the ordering of these co-solutes around the protein, you have a probability of location at a certain radius. Could you infer something about that from this kind of data?
So if I understand, sorry, because there's a pause, I couldn't hear completely, but if I understand your question correctly, so can we get more insight about like how the effective radius of the interaction?
So I guess sorry if I'm being unclear, the ordering, if you had some kind of effective order parameter for how the co-solutes are organized around the protein, it would seem you have, it's not uniform in the blue case, and of course for the unfolded case, it's more uniform, but is that something you could actually back out of your data as a model?
So yeah, I kind of think about similar thing, but for now, I don't know the answer to these questions because I mean, even if I know all the information from NMR, what we can say is only a little bit. We have to combine. So basically, my answer to this question is from the NMR itself, what we can interpret is limited. But if you combine with, for example, molecular dynamic simulations, then maybe we can get more insight about how the water orders and all this stuff. But with NMR by itself, right now I don't know how to get this kind of information. So
You're
yeah.
speaking to the converter about combining with other methods. So I think this has implications also for any in-cell work also, right? Because
Yeah.
then you're looking at other kinds of interactions with proteins as well. All right.
Yeah.
Well, let's thank Yusuke again for a fascinating talk.
Thank you very much.
And so we're continuing on. So our next speaker is Subrata Mishra, and coming from Johns Hopkins, I believe. And I think we're going to hear about large-scale molecular events, possibly.
Can you all hear me? Good. Thank you all for being here. My name is Subrata Mishra, and I'm presenting my work from Dominic Froh's lab at Johns Hopkins. A quick disclaimer though, my current affiliation has no bearing on this work, and there's no conflict of interest. So welcome to the world of non-ribosomal peptides. If you thought you didn't know much about it, look into your medicine cabinet, take out that Neosporin. The active compound is this first one, bacitracin. The second one, I hope you haven't interacted because it's the causative agent of the plague. This third one is an anti-cancer drug, and there's more. You may have heard of penicillin. But the common thing that binds them all are these red, which basically are the peptide bonds. And also you have heterocyclization, which creates more chemical diversity in terms of creating oxazolene and thiazolene rings by cyclodehydration. As diverse as these non-ribosomal peptides are, they are made in a very standard assembly-like manner. And what you have is these massive mega-Dalton enzymatic systems called, not surprisingly, non-ribosomal peptide synthetases, and I'm going to refer to them as NRPSs. You have these massive enzymatic systems that have domains and modules that do the work. And so basically what happens is a module is this bunch of 3 core proteins, domains that you need, and a module will grow the peptide chain by a single substrate. So this squiggly line you see is on this protein and a domain called a carrier protein, and those are like way stations, and these are phosphopentethylene arms, and you have the substrate that is loaded by the A domain, that's the adenylation domain using ATP, and then you have the condensation domains, the C domains, condense the product and the chain elongates, it goes to the last module, it's released, microcyclized. Now, since this works in such an assembly-like manner, what is really attractive is if you could replace a whole module or replace a whole domain. But to be able to do that, you have to understand how chemical synthesis and the interaction between the domains work. Now, I know you saw in this animation that these substrates were levitating. That doesn't happen. There's no diffusion, nor are they playing lacrosse. What does happen is that these carrier proteins do visit the catalytic domains. And that's been seen in negative stain images and more recently in crystal structures. But there seems to be no correlation between the chemical synthesis and the crystal structures. What I mean by that is these 4 conformations that I'm showing you, they're not at different stages of chemical synthesis. They are basically 4 conformations that were fished out from 10,000 crystallization trials. So it seems the only answer we got from these massive, and great work by the way, is that there is interdomain flexibility. Great. What is even more frustrating is that domains don't seem to show conformational change when they interact with other domains. So what you're seeing here is a cyclization domain which does condensation and heterocyclization from a cryo-EM structure. And I've only extracted the heterocyclization domain, and it interacts with the upstream carrier protein, and then it interacts with the downstream carrier protein, and it interacts when the substrate has been passed on. But there is not much of a conformational change when I put, overlay all three of them, which is also not a surprising trend. This has been seen for the last 10 years. What you see in these 4 quadrants are different NRPSs, and as they go through interacting with their other domains, whether there's a substrate present, whether there's a phosphopentaphenyl arm present, there doesn't seem to be much of a conformational change. So we figured since you do the single atom technique called NMR, perhaps we can provide a better perspective and solution to what's happening here. The system we work on is the Yersinia bactin synthetase, and I specifically will be talking about this CY1, which is a condensation and a heterocyclization domain, and its interaction with the upstream carrier protein ARCP. So what CY1 does is condenses the salicylate on the upstream and the cysteine on the downstream, and then it cyclizes the cysteine to a thiazolene, and then there are other reactions and there are other domains involved, and finally you get Yersinia bactin, which is a siderophore, and the causative agent of the plague. So again, I'll be talking about CY1. So the first thing we did is we determined the structure of a 52 kilodalton CY1 protein. It's about 450 amino acids, so it was no small task. But I'm not going to talk about that. A lot of method development went into generating that structure, and we can talk about it later. But what I want you to take away from this slide is that there was conformational heterogeneity in the structure bundle. And the structure itself, what I want you to remember going forward is the one on the left is a C-term subdomain. They have like 2 pseudo subdomains, and one on the right, light blue, is the N-term subdomain. The one in pink is where the upstream carrier protein would bind, and the one in purple is where the downstream carrier protein would bind. Now, you also see some sort of tunnel right there, and I'll come to that in a second, and there's some heterogeneity in access to that tunnel. But this tunnel hypothesis has been seen, this tunnel has been seen in other crystal structures, but this tunnel hypothesis of accessing the active site, which by the way, is approximately center of this protein, has some issues. If you remember my first slide, those NRP peptides can get pretty long. So if they have to access the active site, it seems what is being suggested is that as you go down the module with longer and longer chains, you have to thread in, transfer your substrate, and thread out. But condensation domains don't get bigger as you go down the module, neither does the tunnel. So and this conformational heterogeneity here seems to suggest maybe that's not how it works. Maybe there's some kind of gating that's available for these substrates to access the active site. So we did relaxation dispersion experiments, and we find that there is widespread dynamics in this CY1 protein. And the first thing we did is, since we had excised this domain from the module, we figured, oh, maybe we are accessing a higher energy state that's unfolded. Well, that wasn't the case. What you see on the right in this graph is the difference in chemical shifts from what would happen from random coil chemical shifts and what is predicted for the high energy state by using multi-field relaxation dispersion experiments. This malleability seems to go mostly in the end term. There was 131 residues that showed microsecond, millisecond dynamics, all the way to the downstream carrier protein binding and through the tunnel. What is reminiscent of, though, is conformations observed in crystal structures. I want to clarify here, though. The previous crystal structures I showed you, they were the same NRPS overlaid over each other. These are 4 different condensation domains from 4 different NRPSs that have crystallized in 4 different conformations. So this end-term microsecond, millisecond dynamics that we see seems to be a mirror of that. But is it there for something? Is it there to tell us anything about mechanism? Before we answer that, we wanted to see if and how CY1 interacts with the carrier protein if the loading of the substrate makes a difference. So we did differential isotopic labeling, and indeed we could follow it. So what you see here is this spear in pink is the substrate being loaded, And we have a marker which shows us the substrate being loaded. And what you see on the right, these 5 blocks, they are markers on the CY1 to show a minor conformation emerging. So we found a correlation between the loading of the substrate on the carrier protein and how it engages with CY1. What we also saw is that unless there's a substrate on the carrier protein, it does not engage with CY1. And what you're seeing here are IDIS experiments to monitor the carrier protein. And this is actually a 2D projection of a 3D HNCO to show minor signals that emerge for the other conformation. And what might seem like a banal titration description of an experiment has immense implications on NRPS. And why? Because if you don't have a substrate, ARCP does not interact with CY1. We also did the reverse. Once we loaded a substrate, we use a thioestrase to unload it, and again, the signals go away. And the reason it's important, it directly focuses on the tunnel hypothesis I talked about before. If indeed the tunnel is the way to access the active site, then the carrier protein with its arm, without the substrate, should have been able to access the active site. But that's not what happens. So there seems to be some kind of gating, and that gating is dependent on that substrate loaded on the carrier protein. So we mapped all the residues that were showing a minor conformation, and that too maps along the path that connects the upstream carrier protein and the downstream carrier protein and along the tunnel and the interior of the tunnel. And basically it says that the malleability we saw and the interaction of the carrier protein with the substrate loaded might have some correlation. There might be some kind of flexible gating. So we know now what the reporters are for the interaction. We use mutagenesis to figure out if we could just take away the malleability, and then we could ask the same question, is the malleability really important for anything? So we chose a mutant that's roughly halfway between the 2 interaction sites of the carrier proteins. And this mutation is on an aspartate that is conserved in heterocyclization domains. And it has been shown in many NRPS systems that it takes away the amount of heterocyclization or condensation. If this were a static protein, that mutation should have local effects in the chemical shift perturbation. That's not what happens either. This single mutation has chemical shift effects that propagate all the way from the mutation to both sides where the carrier proteins would interact. Moreover, this mutation stabilizes the wild type by 5 degrees seen from the thermal melts, and it becomes more rigid. And then rigidity is all throughout. It's a global rigidity. What you see on the top in the yellow spheres are where the relaxation contributions from chemical exchange have reduced. There's some increases, but they are few and far in between. So now we have a mutant that is not as dynamic. The flexibility is gone. So now what? Does the loading of the substrate on ARCP do the same thing? It does not. The intense amount of signals, of the minor signals that we saw for the minor conformer earlier for the wild type are gone. It's not completely gone. You barely see like 5 signals, and they are much lower intensity, which is kind of a good thing because it says the 2 still interact, but the flexible gating is gone, even though the ticket for the flexible gating, that is the substrate attached to the carrier protein, is still there. So what does that tell? How does it work in the bigger scheme of things? Well, there has to be some method to the madness if you're having so many modules, and at the end module is going to release your substrate, right? So what this is telling you is the intra-domain dynamics is the flexible gating for the upstream carrier protein, meaning if there is no substrate, it will not go to the active site. And that has implications, because as you keep growing the chain, remember what is downstream for one is upstream for the next. So that has to, so there is now a directionality to this peptide chain growth. And that, when you take away the dynamics, the flexible gating is gone. You can have a ticket, but nobody's going to allow you to enter. And that's how intra-domain dynamics choreographs inter-domain dynamics. Now the D391 itself is quite intriguing. One single mutation creates so much havoc, or rather takes away the havoc. And it seems this D391 is located at a place that has a high electrostatic negative potential. So I figured maybe the reason the dynamics goes away is because in the wild type, the relief of the electrostatic negative potential allows access to more conformations. So you take these 391 out, you don't have access to as many conformations. So I wanted to show it in a more general way though. So what I did is I took consensus sequences of these heterocyclization domains. I aligned them and threaded them through the 4 conformations for the crystal structure. So it's like a poor man's molecular dynamics, right? And then I got the same sequence on 4 different conformations. And indeed, there's a conformational dependent relief of electrostatic potential. I took it one step further. There are many subtypes of condensation domains. They all have slightly different functions, and we can talk about that later, but they do one thing in common. They all have to allow carrier proteins with their phosphopentathionyl arms, with their substrates, access to the active site. And what you see in this grid is on each row, it's the same sequence with 4 different conformations. Each column is the same conformation with 5 different sequences. This grid represents the information from 525 proteins. And what it's trying to tell you is that there is a conformational dependent electrostatic potential relief, but it's not always the same conformation that offers it. So what is the message I want you to go home with? It's that you can have the same structural fold, but you can fine-tune the malleability to produce different function. And that's pretty powerful in terms of understanding evolution, in terms of understanding mutations you make and the results you get. And with that, I come to the end of my talk. All this work would not have been possible without Dominique's constant support through thick and thin. And to allow me to end the story, Aswani worked with me on the structure determination. Kenny was a graduate student I recruited, and he did some fantastic work on the mutants. He's going to be out, graduate next year, so look out for his applications for postdoctoral positions. Ananya, great help with NMR experimental setup. Our collaborators, Nick and Santrupti, who helped with the CS Rosetta for the structure determination. Guillaume for Chemex and helped with the RD data analysis. Dan gave us a structure of a different NRPS heterocyclization domain. And finally, David Weber for his generous use of the 950 data for the RD-CPMG experiments and for a timeshare Trosi-Nosy that I designed for structure calculation. Lastly, NIH for the money and a bit of shameless plugging. I'm in the job market, so if you want to know what my future research plans are, come talk to me. Thank you.
Beautiful talk, Subrata. It underscores how nature can take a limited number of templates, but get diverse functionality from a limited number of.
I'm sorry, I missed that first part.
I think you're showing, at least you're showing me how you have a limit, you can have a limited number of folds.
Right.
And yet you can have so many more functions.
That's correct.
Yeah.
I mean, you see a lot of kinases, phosphatases, transferases having the same structure and again. And you know, they may be orthologs and they might have slight differentiation of function, but they have something in common from which they diverge. And malleability or the internal dynamics may be that something.
Fantastic. Go ahead.
Yeah,
Yes.
yeah, very impressive study, like 52 kilodalton protein,
Thank you.
and
Thank you.
beautiful data of relaxation dispersion. And but typically, this kind of enzyme protein complex, there is surfactant analog which stabilizes a complex and makes structural analysis easier. In this system, did you find any analog which makes a complex stable, or
Analog that makes a complex stable? Could you expand on that? Did you mean like a different substrate on the carrier protein?
yeah, substrate and carrier protein, and that complex lifetime become much longer?
I see. Almost like you're saying, like, let's search for the transition state analog. If I did that, I would be rich. But thank you for the question. I really appreciate that. I think no, the real answer is no, but these substrates on these phosphopentathionyl arms also gets hydrolyzed. So non-hydrolyzable analogs have been made for this carrier protein, and Andrew actually worked on this carrier protein and making these non-hydrolyzable intermediates attached to the PPANT arm.
That may be interesting.
Absolutely,
Yeah.
absolutely.
Yeah, thank you very much.
Thank you very much.
Other questions? I have one is, when you show these chemical shift perturbations from the mutation and they seem to propagate, right? On the one hand, that tells us, okay, something interesting is going on, but then if one tries to go further and have an interpretation of this in terms of conformational perturbations, there's a lot of pushback. Right? Like we don't know anything what this chemical shift perturbation means. Do you have a sense of what, from all those structural comparisons, what the physical basis for those widespread chemical shifts might be?
Right, right, and that's a very good question because it's a question that attacks directly the heart of the matter because we consider structure and dynamics two different things. But dynamics is a way to access more conformation. So you have to analyze the data as a whole rather than just seeing the chemical shift perturbation. Because obviously, I think what you may be hinting at is chemical shift perturbation a complete change in structure? But what I would say there is that in the context of the data that the contribution to REX reduced from chemical exchange, the overall context tells us that we have access to less conformations and that chemical shift is now averaging in a different way. And that is the CSP that we are really seeing.
All right. Well, thanks again.
Thank you.
So our next speaker is Giuliano Antelo from, I guess I'm going to mispronounce this, so I will let you show on your slide. It's not advancing.
Our previous talk. I'm here to talk about the role of conformational entropy in the molecular evolution of bacterial transcriptional repressors. My name is Giuliano Antelo. I am supervised by Dr. Diana Capdevila during my PhD. We work at the Leluar Institute in Buenos Aires, Argentina, which is named after the Argentinian Nobel Prize, Dr. Luis Federico Leluar. And the University of Buenos Aires is where I'm getting my actual degree. So, okay, this is not working. Can I get some help? Sorry. I think it's stuck. But it's not.
Go to the next picture.
Let me open it again.
Oh, that's the previous one.
Yeah. Okay, sorry about that, so sorry. So like I was saying, as a biologist, I am interested in biological problems. And the biological problem that we study in our lab is how bacterial pathogens deal with stresses imposed by their hosts. In particular, micronutrients such as transition metals and reactive species are essential to any living organism but are toxic in high concentrations. And in particular, the human host affects the bioavailability of these nutrients to combat infections. The so-called nutritional immunity response involves, in part, the secretion of toxic metal concentrations or reactive nitrogen, oxygen, or sulfur species to try to intoxicate bacteria. But successful bacterial pathogens have evolved ways of overcoming this response that are ultimately modulated by allosteric transcriptional repressors. And the way that these proteins work is they sense an inducer either by binding to a metal ion or by doing chemistry with a reactive species, which causes the repression from the DNA, turning on transcription of proteins that will try to keep homeostasis in these stressful conditions. So the family of proteins that we are most interested in is the RSARS family of transcriptional repressors, which are key to the bacterial response to stress. Now, these proteins are characterized by a highly conserved three-dimensional structure across the whole family, upon which 5 different inducer binding sites have evolved in different members of the family with affinity to different types of inducer, either metal ions or reactive species. But what I want to point out here is that all these different inducer binding sites have evolved in distant sites of the molecules to the DNA binding site of the protein, which is located between this helix 4 and helix 3 in this helix-turn-helix motif. Now, allostery has been historically explained in terms of conformational changes, where a binding event induces a conformational change that affects the probability of a second binding event from occurring at a distant site of the molecule. So one question that we asked ourselves is, how did all the different allosteric connections evolve in the same structural scaffold? When we looked into this in a protein called CCR8 from the pathogen Staphylococcus aureus, which is a zinc binding sensor, and we solved the structures of the apo and the zinc bound form, we saw that both structures are highly similar, both for the DNA compatible form, which is the apo form of the protein, or the DNA bound form. So there are no conformational changes that can explain allostery in this protein. And this has not only been observed in CCR8, but for the rest of the proteins within the family that we have the structures, or that other people have solved the structures of the DNA binding competent form of the protein and the incompetent form of the protein, they are highly similar. So the structure alone cannot account for the allosteric coupling between the DNA binding sites and the inducer binding sites in the SARS. So the question is then, well, then what does? So allostery is an intrinsically thermodynamic phenomenon that is defined by a coupling free energy given by the change, again, in affinity for a ligand, given that another binding event has occurred at a distant site of the molecule. So every free energy has contributions coming from enthalpy changes and entropic changes, and the entropic contribution can be subdivided into different entropic contributions, and we are most interested in the contribution of the conformational change, the conformational entropy change in the protein, which is accessible through NMR relaxation parameters. So how do we quantify conformational entropy? We use methyl groups from the side chains of isoleucine, leucine, valine, alanine, or methionine residues as dynamical proxies to access conformational entropy. And the way we do that is through a series of experiments that measure the so-called allowed or simple quantum transitions and the forbidden, or which are the double or triple quantum transitions of the coupled spin nuclei in a methyl group. And the ratio of intensities between those two measurements allow for the extraction of the so-called axial order parameter, which is this S-squared term that can have values ranging from 0 if the side chain is completely flexible and 1 if it is completely rigid. So again, what we are doing is using methyls inside the protein as dynamical reporters of the conformational entropy, because it has been shown that the average change in this axial order parameter is proportional to the change in the conformational entropy of a protein between two states. So what happens in our system? In the case of CCR8, the protein that I was talking to you about earlier, we saw that zinc binding induces a global rigidification of the protein, while DNA binding induces a flexibilization of the protein when it's bound to the DNA, which is something that you wouldn't intuitively expect when you are binding two molecules together. This is what we call an entropy reservoir, and it's quite significant because it accounts for 1/3 of the total free energy of the binding event. So this allowed us to come up with a model for the evolution of these different allosteric connections in which an entropy reservoir stabilizes the DNA-bound form of the repressor, and inducing binding simply prevents access to this reservoir. But to test this hypothesis, we need to know what happens in the rest of the family, not just one member. So what do we know about the rest of the family? And the answer to that question is fairly little. What I'm showing you here is a sequence similarity network in which all the proteins that are annotated in UniProt as SARS are represented in these nodes, and the lines connecting nodes represent a shared 40% identity between the proteins in each node. What do we gain from this? Well, when we threw the information of the R-stars that have been characterized by others in the literature, which are these black stars that are scattered around, we saw that sequence similarity segregates the family according to function, okay? And what is interesting is that the two most, the two most characterized clusters in this family are these cadmium, zinc, and cobalt sensors, where we find CCRA, and a second cluster, which is highly biochemically characterized, is one corresponding to reactive sulfur species sensors. So the strategy that we took is try to see if in these distantly related proteins from these clusters, we could also measure this entropy reservoir. So the protein that I'm working with is called SQRR, which is a master regulator of reactive sulfur species. The way that the protein works is that in presence of reactive sulfur species, the reactive cysteines in the protein become oxidized, forming a tetrasulfur linkage that causes the repression from the DNA. Here I'm showing you the structure of said tetrasulfur linkage. And again, when we solved these structures, we saw that the reduced form of SQRR, which is DNA compatible, and the tetrasulfur form of SQRR, which is DNA incompatible, share a highly similar three-dimensional crystal structure. So we wonder on the role of conformational dynamics here. And in order to assess that question, we need to characterize the states on which this protein can be found. So starting from the reduced to tetrasulfur transition, we saw that we were able to acquire high quality spectra for both states. The standard three-dimensional spectra allow for the almost complete assignment of each state. And analyzing the chemical shift perturbations, we saw that they are mainly localized around the reactive cysteines of the protein. And they are actually less than the ones that we would have expected, predicting them from structure. So again, there are no significant big conformational changes in this protein. And when we looked into the fast internal dynamics, again, we obtained very high quality spectra for both the reduced and the tetrasulfur stage. We saw that tetrasulfur formation freezes the internal dynamics of the protein with a huge entropic penalty involved, which is reminiscent to what we had observed in the other protein that I told you about, CCRA. So is there an entropy reservoir also driving DNA interaction here? So then characterizing this process, working with DNA complexes in NMR is not trivial. In particular, these proteins are homodimeric. And in order to prevent doubling the amount of signals that we measure in NMR, we need to work with a palindromic DNA. The problem is that the natural operators that this protein binds to are not palindromic, so we have to design one based on the natural sequences and try to make it as short as possible to keep the size of the particle as small as possible. So we characterize the interaction for this synthetic short palindromic DNA. We saw that it retained high affinity for our molecule, so we carry on doing experiments. And again, we were able, here I'm showing you in green, the spectra for the DNA bound complex. We were able to obtain both for the side chains and for the backbone high quality spectra. I do not have an assignment yet, I'm working on it, but nevertheless, I can extract the dynamical parameters needed to quantify the changing conformational entropy of the complex. The way we did that, we estimated the correlation time using the TROSY for rotational correlation times or track pull sequence instead of the typical T1, T2 approach, because this is more appropriate, the track pull sequence is more appropriate for large size particle because it's just a series of 1Ds that you integrate through time. And this is the correlation time that we got, which is in accordance to what we expected for a particle this size and another protein DNA bound complex that we have worked with in the past. And again, given the size of the complex, the higher sensitivity of the triple quantum experiments allow for a better estimation of the axial order parameter than the double quantum. And we can see that in the fits for the peak ratio between the forbidden and the allowed transitions, the triple quantum experiments show a much better fit than the double quantum experiments because those spectra were much more noisy. So what we got was this value for the axial order parameter, the average axial order parameter for the DNA bound complex, 0.3544. But to be academically honest, we need to take this value with a grain of salt because we would expect that the axial order parameter for the alanines, which have few degrees of freedom, are around 0.8, have a value of around 0.8, and we are not seeing any residue with those values. So we are working on trying to understand why this might be happening. If anyone has any comments, please do so. But if we take that value, we would be in the presence of, again, an entropy reservoir driving, at least in part, the DNA interaction and affecting this process with a favorable entropy contribution. Bringing back CCRH as for a comparison, I told you that the change in conformational entropy accounted for 1/3 of the total free energy of the DNA bound complex. And when we measure the thermodynamics in SQRR using ITC, we were shocked by a huge enthalpic contribution driving this process. We cannot neither explain this at the moment. We think it has to do with different binding modes that this protein has for their operator that are different from the ones that CCRA has. But I would like to show you something regarding to that, but for the sake of time, I'm just moving on. So we were able to characterize the reduced and tetrasulfide transition, and we are working on validating the results that we observed from the reduced to DNA bound complex formation. So to sum up, despite its central role in pathogenesis, the RSARS family of transcriptional repressors remains largely uncharacterized, as I'm showing you in the sequence similarity network. In at least one member of the family, CCRA, an entropy reservoir is driving DNA interaction, and inducer binding prevents access to this reservoir, giving rise to the regulatory function of this repressor. In SQRR, which is a functionally distant homologue, the contribution of the conformational dynamics to its regulatory function seems to follow this same trends. We are working on validating these observations and expanding our knowledge on the fundamental mechanisms seized by evolution to affect protein function and give rise to new forms of bacterial resistance. With this, I would like to thank everybody in the audience. My supervisor, Dr. Diana Capdevila. I would like to acknowledge the work done in these projects by Christian Pizies. We shared the same contribution to this project. To thank Professor Guidriok, who kindly hosted me last year in his lab. And I would like to thank the ANC for granting me the travel stipend that allowed me to come here and present for the very first time in front of an audience part of my research. I believe that as any human activity, science is a social activity, and it makes sense because we communicate it both with our peers and with the general public. So thank you, ANC, for allowing this opportunity, and thank you.
Thanks very much, Giuliano. I have a quick question. Is what do you think this is? What's your speculation as far as new avenues to combat antibiotic resistance? What does this suggest? Maybe new strategies to combat resistance.
Could you repeat? I didn't get that.
To combat antibiotic resistance, does your results, which you care to speculate, does it suggest new strategies for combating?
Yes, exactly. That's the like in a long run, we would like to. So to understand how these proteins work will benefit us from trying to understand how new forms of bacterial resistance might emerge, and in particular, the diversification of this protein family is what has allowed new forms of bacterial resistance to emerge, and understanding whether or not there is a more delocalized property of the protein that allows for its regulatory function. That would be this entropy reservoir. Would be I don't want to sound like, but like beneficial to evolution in the sense that there is more it's more evolvable another allosteric binding site in a molecule when you don't have to wait for allosteric connections, physical allosteric connections between the residues of one binding site and the other binding site. So that's.
Yeah, thanks. Very interesting story.
Thank you.
It's a very basic point. I mean, you have the two two types of proteins. The site is the same. There are cysteines which are either binding a metal ion or get oxidized.
Yes.
And from the electrostatic point of view, so would be the enthalpy aspect and not the entropy, you have a different situation because the charges are different. In one case, you lose 2 protons and to get the disulfide bond form. And in the other, you lose 2 protons, but it's a 2+ ion coming in. So the electrostatic is different. So you should have an enthalpic difference in the energetics of the region around the metal binding site of the disulfide bond. I don't know if you, I mean, beside the entropic aspect, but overall the free energy, there, I would guess there is quite different enthalpic contribution. I don't know if you have analyzed this aspect.
That's quite interesting. Yes, we have analyzed the other contributions for CCRA. We haven't done this in SQRR. We have looked at the thermodynamics that have been reported by others in sensors that are similar to SQRR in the sense that they have also reactor systems that react with sulfur species and perform their function. And we do not see in them an enthalpy term as big as the one we are seeing here. So it might be adding to that in the sense of what we see in CCRA and what we see in this other molecule, but it also doesn't correspond with more closely related ulcers to
Is a fact that in one case you have a 2+ charge.
Right,
more.
yeah, no,
So
you are absolutely
something
right.
might be there are locally, in some cases it happens that some charged residues slightly move. So if you look at the backbone, you don't see any change, but you might look at some residues, so let's say lysine, that they move back and forth, for example, that it compensates a change in charge. That is a local effect that might be relevant.
That's interesting. Yeah, I will definitely look into it.
Somehow the answer is always water, right? In a biochemistry exam, it's all water, but I'm just kidding. Thank you very much.
Thank you.
Kenneth. Thank you very much. Okay, our last speaker is Junji Uehara, coming from the University of Texas Medical Branch.
First of all, I would like to thank the ENC organizers for the invitation. Today, I'm going to talk about biomolecular electrostatics by NMR. Probably many of you in this audience are in the field of structural biology or molecular biophysics. And when we discuss electrostatic potentials, usually we use this kind of surface representation of the electrostatic potential. So I got cursor here. But today I'm going to talk about basically 3-dimensional electrostatic potentials around the protein. You can easily calculate this kind of potentials using nice software like APPS or Delphi. And those software solve Poisson-Boltzmann equations, and you can easily get electrostatic potentials from just structure. That's very convenient, but you have to remember that Poisson-Boltzmann theory is approximate with some known limitations. For example, you may not get very accurate electrostatic potentials for zones near the hydration layer, or for the system highly charged like nucleic acid. And the accuracy is limited at high ionic strength. But the problem is that the validity range of the Poisson-Boltzmann theory is not well defined because the experimental studies of electrostatic potentials are really difficult. The more serious problem arises from basically dynamics. Because of the protein dynamics, charge positions are changing constantly in solution. For example, the charge at the tip of the side chains are moving due to side chain dynamics. And there are many loops, and some proteins have multi-domains connected by linkers, and those linkers can be quite flexible, and the relative positioning of those domains can be quite different. So because of this kind of dynamics, the computation of electrostatic potentials from structures could be pretty much meaningless, especially for something like intrinsically disordered proteins or intrinsically disordered regions. So it would be wonderful if we could measure electrostatic potentials without using any structural information by NMR. Question is it possible? The goal in my talk today is to demonstrate that the answer to this question is absolutely yes. The method I'm going to introduce depends on solvent PRE arising from charged paramagnetic co-solutes shown here. These are nitroxide with charged moieties. And if you put either one of them in your protein solution, the spatial distribution of these co-solute molecules are biased by electrostatic potentials. And such bias are different for these two compounds because their charges are different. And by analyzing PRE for these co-solutes, basically, we can get effective near-surface electrostatic potentials. In the next few slides, I will explain how it works. The most important equation for that is shown here. This was published by the group of Marius Kloa, my former mentor. Actually, this equation has been already shown in Dr. Okuno's presentation. Here in this equation, R is the distance between the paramagnetic center and observed nucleus, and U here is the potential mean force for a particular position. If you convert this equation into 3D lattice space, it becomes this relatively familiar form, which is quite similar to Otting-Lamaster equation. But there is a fundamental difference in that it contains Boltzmann factor that represents statistical weight according to the potential. And when we measure PRE for these two co-solute molecules and take a ratio, the ratio becomes in this form, and these terms can be canceled, assuming that assuming that the correlation time is identical for these two. And this ratio is governed by the co-solute molecules in a space very close to the observed nucleus. And in that relatively small space, the potential could be quite similar. And in such a case, we can approximate this quantity by this one. And here, the delta U is a difference in potentials. And this potential could have both electrostatic component and non-electrostatic component. For example, for any nitroxide, it's known that they have some hydrophobicity. So there could be a hydrophobic bias in spatial distribution. But such effects are canceled because for these two compounds, hydrophobicity are pretty much the same. So the hydrophobic effects are canceled, but electrostatic component remains. So this one can be approximated by this one. And by connecting these two, we can define effective near-surface electrostatic potential shown here. And here, this gamma-2 ratio is from the experimental data. Okay. Good thing about this phi NS electrostatic potential is that this can be back calculated from the structure using this equation. Okay, so here, this R is the distance between grid point and observed nucleus, and phi I is the electrostatic potential from Poisson-Boltzmann theory. So using this equation for the entire space of the grid space, you can back calculate phi ENS, and you can compare the experimental potential and computational potential. Okay, you might be wondering, what is really the physical meaning of phi ENS? The physical meaning of phi ENS is relatively simple. So basically, as I mentioned, it is related to the electrostatic potentials near the observed nucleus. And in fact, empirically, if we define a zone that makes 68% contribution to overall or sum of R2 minus 6, the average of the electrostatic potentials within that zone is almost the same as the back calculated ENS calculated by the previous equation I showed. Okay, here is the pulse sequence we use for the measurement of PRE rates. I mean, transverse proton PRE rates. And here we use 3 samples. One is just a protein solution, and the other 2 are with co-solute molecules as shown here. And this is PRE data measured for ubiquitin using either this or this proxial derivatives. And here, gamma-2 values are quite different for each residues because the electrostatic bias for these 2 are quite different due to different charges. Okay, from the gamma-2 data, we calculated the electrostatic potentials using the definition of phi ENS, and red data points are the experimental phi ENS values. We also calculated the same, corresponding phi ENS values using a Poisson-Boltzmann theory, and the blue line shows those back calculated phi ENS. And green is basically a simplistic approach using a particular cutoff for the proximal zone around each proton nucleus, and basically it is just average of that. As you can see, the experimental phi ENS values are quite close to the calculated electrostatic potentials. And if you compare them in the correlation plot, it's like this, okay? The agreement between experimental and theoretical electrostatic potentials are quite good, especially for secondary structure regions. But you see some large deviation for loop regions. And we thought that this could be due to basically mobility. And to test that idea, we generated 100 structures. And for each structure, we calculated phi ENS for individual residues. And here, the green bars shows average and standard deviation of such calculated phi ENS values. As you can see, some of the deviations can be explained by this kind of structural flexibility. We also tested this methodology for methyl groups. Excuse me. As you can see here, the agreement between experimental and theoretical electrostatic potentials were quite good for methyl groups as well. This is important because, as you know, methyl groups are quite useful for animal studies on large molecular systems. So probably this methodology will work even for relatively large systems. We also applied our method to a highly charged protein DNA complex. In this complex, the protein antennapedia homeodomain is highly negatively charged. However, highly positively charged. However, because of even larger negative charge of DNA, the electrostatic potentials from the experiment clearly shows that all of the residue shows negative electrostatic potentials. This reflects basically a strong influence of the, electrostatic influence of the DNA. And agreement between experimental potential and theoretical potential were quite good for secondary structure regions, but not for the loop region, as we saw for the ubiquity. This methodology can be applied for some different things. And here I just show 4 examples, which we are actually working on. We can use this methodology to examine the validity range of theoretical electrostatic models. We can also use this method to assign NMR signals based on electrostatic potentials. But more interesting application would be for disordered proteins, because for disordered proteins, as I said earlier, it's very difficult to get electrostatic properties from experimental data. But now, with this methodology, we can get electrostatic potentials without any structural information. So we can get, we can learn the electrostatic properties of IDPs or IDRs. And we can use such experimental electrostatic potentials to get a reasonable, electrostatically reasonable structure ensemble of IDPs or IDRs. And we are now trying to do that for HMGB1, and I'm going to show some preliminary data on that. HMGB1 is a 215 residue protein, and the C-terminal 30 residue is completely negatively charged, either glutamate or aspartate. And it has 2 DNA binding domains that are positively charged. And because of those opposite charges, these 2 segments, DNA binding segment and C-terminal segment electrostatically interact, and it causes autoinhibition. But this effect is dependent on ionic strength, and at very high ionic strength, autoinhibition is released, but at physiological ionic strength, more than 90% of the protein is basically auto-inhibited due to the electrostatic interaction between these 2. But this auto-inhibition doesn't cause static structure. Even within the auto-inhibited state, the structures are quite dynamic, and we would like to know quite reasonable, electrostatically reasonable structure ensemble for this system. And for that, we used phi-NS method, and these are data for the full-length HMGB1 and the truncation mutant that doesn't have the negatively charged C-terminal tail. As you can see here, just like what we saw for Antennapedia DNA complex, due to the strong negative charge of the C-terminal tail, we see quite a drastic shift of the electrostatic potentials around DNA binding domains. So this is really showing electrostatic influence of the C-terminal tail. So in principle, we can use this methodology to get a reasonable ensemble for the highly dynamic system like this one. Okay, this summarizes what I have talked so far. This phi-ENS methodology is a unique, powerful, and simple method to measure local electrostatic potentials. And you can get the electrostatic potentials just from relatively simple PR experiments on 3 samples. And you don't have to do any chemical modifications for this method. And phi-ENS can be back calculated from structure. And this is particularly useful for investigations of dynamic systems like IDPs or IDRs. Here, I compare our method with existing other biophysical methods for electrostatic potentials or electrostatic fields. Among them, the most commonly used method is pKa shifts. But pKa shifts do not directly provide electrostatic potentials. And also, the methodology of pKa shift is limited to titratable groups. And you have to use a wide range of pH, like pH 2 or pH 12, at which proteins could be denatured. On the other hand, our method can be done at a single pH, and it can be, it can give electrostatic potential directly for many proton nuclei, including amide and H-alpha or methyl groups. So our method has major advantage over pKa-based assessment of electrostatic models. Stark shift in vibrational spectroscopy is quite powerful, and this is extensively used by Steve Boxer's group at Stanford. But the major drawback of this method is that it still requires chemical modifications. Same thing is true for double resonance EPR or redox potential-based assessment of electrostatic potentials. They all require chemical modifications, but our methods do not. So our method have major advantages over other methods for electrostatic potentials. With that, we believe that this methodology adds a unique capability to NMR spectroscopy. Here is the people who contributed to this work. Behind you made major contribution to this work, and we got a lot of theoretical guidance from Monty Pettit. And these are our funding source. And finally, I thank you for your attention.
That's great, Genji.
It really gets at a basic interaction, but in a very efficient way. Just a quick, maybe it's too early, but with the first talk we heard, talking about the cell environment, pH varies in different parts of the cell. So in principle, you could use this tool at some point to look at the effects of different pH in different parts of the cell, I suppose. Is that reasonable, or is that too far off?
It might be a little bit too far. Okay. One thing is the two compounds here with amino group and carboxylate group. As you know, the pKa of carboxylate group is around 4.6 or something like that. And for amino group, it is probably around 9 point something, or maybe 10. So if you use something like pH 5, some of the carboxylate becomes basically protonated, and it loses a charge, right? So there is a limitation, in terms of pH range. But a good thing is that this method doesn't really require multiple pH values. A single pH like 7.5 is just fine for this one.
Questions over there?
Hi, pretty ingenious experiment. Thank you for that. I was wondering, these probes that you're using, so the electron spin is not localized at the exact same position as the charge. Does that affect the result in some way? Can you comment on that?
Okay, so basically here, the basic assumption is that the paramagnetic center and the charged moieties are basically occupying the same space. However, that is a little bit simplistic, and some of the deviation we saw turned out to be actually due to such spherical model. Basically, we assume that the paramagnetic center and charge center is at the center of the spherical representation of the paramagnetic group. But more realistically, we should use at least two model representation because those two parts, I mean, charge moiety and paramagnetic centers are at different positions. So we are now trying to improve this method by basically developing a better model and better representation for the paramagnetic co-solute. But that's probably feasible.
Very nice, thanks.
Thank you. That's a very clever application of PREs that you presented there. I'm having two questions. My first would be, the framework that you're showing is, if I'm not mistaken, based off sums of pairs of electrons and nuclei. But when you work with this framework, is it possible that you attract more electron spins towards one nuclei so that the pairwise interactions might not sufficiently explain the effects that you have, for example, interferences between three spins?
Okay, so actually that question is answered in the previous paper by Maris Kluwer's group. And for example, concentration effect of the co-solutes are already taking care of that. So it's not really the issue. And in fact, actually, as Dr. Okuno showed, even at a relatively high concentration, like 25 millimolar of co-solute molecules, we can still get quite reasonable agreement between experimental data and theoretical prediction based on our models. So for this part, probably there is no problem in theory.
That's good to know. My other question would be, are you worried about thermal energy, like the Bjerrum lengths, for example, that might cancel electrostatic interactions? From what I know, between two charges, thermal energy, the distance they need is 0.7 nanometers, so that the thermal energy is the same. So that maybe you don't see some regions of distances because the PRE is probably much longer, the PRE.
Okay, to answer that question, probably this equation is quite useful. So basically, if there are some differences between these two compounds and they are not canceled by this one, such effect may contribute to the observed PRE ratio. But if they are minor, the major contribution should be electrostatic. That is basically a sort of assumption, and this assumption seems to be working pretty well. That's all I can say.
Okay, thank you.
Very interesting work. One question, I have a couple of practical questions is that, what is the concentration of this co-solute that you use? It's probably not compatible with something like a DTT that normally people use in a protein. And the second is, how do you treat water in this case as water becomes broader with these co-solutes?
Okay, so for the concentration, we typically use a concentration between 10 and 30 something, depending on the necessary effect. For example, if you use a very low concentration, the PRE effect becomes too small to use. And to use this methodology, the PRE has to be significantly larger than 0. So you need relatively high concentration to achieve that, especially if you are interested in region deep inside of the molecule. So depending on which region you are interested in, the necessary concentration of the co-solute is different. Sorry, what was the second question? Sorry.
It's not probably compatible with DTT, right?
Oh yes, for DTT, DTT probably reduces the permanent co-solute, I mean nitroxide. So unfortunately, this methodology doesn't work in the presence of DTT or ascorbic acid.
And the water suppression is okay? Does the water line get broader with 10 millimolar of this, or?
There is no problem about water suppression in our experiments. So yeah, of course, the relaxation property of water also changes, but that doesn't interfere in our previous experiments.
Any other questions? If not, let's thank Chunji again and all the speakers for this biomolecular session. Thanks.